So what I'm going to try to do is to give you a blend of my sort of science and how I view at least some of the initial problems in trying to map the structure of the human genome with what I perceive to be your interest, which is what can you do in this project over the next 5 to 15 years. What are the current needs for enhanced sort of computer applications in this area, and what are the perceived future needs? And I'm going to try to jump back and forth between these two. And if I can figure out how to use all this electronics and get the first slide. Okay. It's not yet showing on my screen. That's really an outline. That's an outline of thanks. Yeah, let's get it. The scale is the key word here. Let's scale the slide. That's an outline of what I'm going to do. I want to first just remind you about what the scale of this Human Genome and Human Genome Project is all about. I'll recapitulate a few of the comments that Eric Lander made. And then I'm going to talk about types of maps very briefly, the methodology that we use to make physical maps. Since Eric's talked about genetic maps and a bit about sequencing, I'll concentrate almost totally on this middle ground, which is physical maps. I'll indicate methods. Also, as I go through this, try to give you a feeling for applications. And then really in the second part of the talk, I'll try to address my own private view of what I think the computing needs are in this field and try to illustrate in a few cases the types of problems I think the computer scientists will hopefully think about and perhaps soon solve. Now, the human genome is very big, and I have different analogies which work for different people to try to let people comprehend just how massive the task is. The haploid is 3 billion base pairs. Of course, the diploid is 6 billion base pairs. That would be a typical cell. The DNA molecules that make up that genome, extended end to end, actually measure 6 feet for the diploid, which is about the size of a human. That's a nice coincidence. I like to think in terms of telephone book analogies. I'll come back to that. I can't think in terms of computer disks, but I can imagine what a few hundred telephone books might look like. That's the database. The most significant thing about the human genome is its polymorphism from individual to individual. Although the estimate's not very good, we currently think that of all the 3 billion base pairs in the genome, about 0.1% of the positions are variable throughout the population. Most of these differences are silent, at least in the sense that they don't produce any dramatic deterioration in the quality of life for the individual who has them. They may just determine the size of the earlobe or something like that. But some of these differences obviously result in serious diseases, and those are the ones that are of most interest for us to try to find. Now, what the human genome actually looks like as an entity is shown here. This is a karyotype. It's human chromosomes arrested in metaphase and stained so that you can see them. And this is a real view. Eric Lander showed you a beautiful, idealized view of what these chromosomes look like, but in practice, in most people's hands, this is about the best that you ordinarily do. And in clinical analysis, a skilled individual, a cytogeneticist, can recognize by the pattern of banding each individual chromosome, and at the moment, pretty much by hand, cuts them out of a photograph, pastes them up into an array, and in this case, you can see and I can see if I can figure out how to use this. Let's see. Okay. It's very hard. Now I see the trouble. I can't. There it is. Okay. Yeah. All right. You can see that this individual, is a male because he has both an X and a Y chromosome. And unfortunately, if we go over here, you can see that this individual is not well. This is 3 copies of chromosome 21. This individual has Down syndrome. Now, the difficulty with this type of analysis, which is done routinely in really hundreds of cases a day probably around the world. The difficulty is that it's done by hand, it's subjective, the information content is rather limited. There are about 300 bands in a typical stain preparation like this, and it's rather difficult in many cases to view the picture finely enough or reproducibly enough to be sure of what one is doing. Now, as we gain more and more information about the physical map of the human genome, I believe it's going to be possible to replace this kind of analysis by one that is much more molecular, much more capable of simple automation, and more important, much more informative. And I'll come back later on and sort of sketch how I think in not too many years we'll be able to do this sort of analysis with about 100 times the resolution. That means about 100 times the information content. Now, the other point I want to make before I leave this slide is that as we make maps, which are just pictures of structure, representations, sometimes abstract, sometimes fairly realistic, what we look for in these maps are patterns. We hope that there'll be some images in the maps which will provide clues to function or history, evolution, organization. And there already is a striking pattern in the physical map of the human genome, even seen at this very crude level of resolution. It's these bands. These bands have been seen by cytogeneticists with different types of staining for more than 20 years, and we still do not know, in any case, why we see these bands. We don't know anything about the molecular basis of this banding. We have hints, and some of those hints are becoming fairly concrete, but no proof yet as to what causes this. Now, in studying the human genome, we have a problem in scaling the effort across many orders of magnitude. The whole genome, as I said a moment ago, is 3 gigabases, if we think of a haploid. And we can sort of progressively divide it into finer and finer chunks. But when we do so, the experimental technology that's needed to handle the problem keeps changing. And so what makes the whole Human Genome Project both interesting and complex is that you have to work with a variety of different types of experimental technology in order to be able to acquire information at any finite rate. So at the level of the intact genome, there's been tremendous progress in physical separation of chromosomes, either by fluorescence-activated chromosomes sorting, a true physical separation, or by various tricks which allow you to clone a human chromosome in, let's say, a mouse cell or a hamster cell. And I'll show you how powerful that is later on. The chromosomes themselves can be studied by banding, as you saw on the previous slide, in the microscope, or by in situ hybridization, or at a level of resolution currently of about 10 million base pairs by the kind of genetic mapping that Eric Lander spoke about in the previous talk. Now, where we start to get interested in this problem is as the resolution becomes higher. And one can start with sort of 10 million base pair regions of DNA and try to divide it into 1 million base pair or a 10th million base pair portions by what I call macro restriction mapping, in principle by much finer genetic mapping, or by cloning into yeast artificial chromosomes, YAC cloning, a very new technique, but one that looks like it has considerable power. In turn, a YAC clone of about ideally 1 million base pairs, today really only a couple 100,000 base pairs, could be divided by finer subcloning procedures, let's say into cosmids or into phage. Each, let's say, cosmid clone might represent only 50,000 base pairs of the human genome. It, in turn, could be subcloned into about 1 kilobase pair plasmids, and those essentially can be sequenced almost in one shot by current technology. Now, that's an outline of what one has to do in what's called a top-down approach to try to make a complete map of the human genome. And again, the difficulty with thinking about this is that we've really jumped, if you consider a diploid, from almost 10 gigabases down to a single base. We've jumped through 11 orders of magnitude in trying to map out this project. And that is such a large range of techniques and scale that I think it's mind-boggling. And I want to try to give you an example to prove to you that in trying to even think about such a wide range of scales and resolutions that you very easily can get lost. So this is a pictorial view of the whole human genome. What you're actually looking at is the Earth from fairly far away in outer space. But please don't zoom. You'll ruin the effect. This is from a fantastic Scientific American book called Powers of 10, which I'm sure many of you have seen. So this is the whole genome. And if we just imagine taking the genome and dividing it up into a series of the best YAC libraries that we could possibly create, a sort of 1 megabase scale, or 100,000 base pair scale, sort of a really fine library, or the ultimate genetic map, almost a map of almost inconceivable power, if we could have a genetic map with 100,000 base pair resolution. Then, instead of seeing the whole Earth, what you're seeing is this picture, which turns out to be an aerial view of part of the city of Chicago. And you're looking here at, that's actually a marina. Those are boats sitting in a marina, I guess part of Lake Michigan. This is a parking lot. And on this level of resolution, there are certain patterns. You can tell that all the boats are in the marina. That's a pattern. This might correspond to a chromosome band. All the cars are in the parking lot. There's no confusion. There were absolutely no cars in the marina. Okay, and there are no boats in the parking lot, I hope. All right, so there are patterns on this scale, but you couldn't conceive of this scale if you started simply thinking about the previous slide, which is the sort of whole Earth, the whole genome. And if we go from this scale, which is sort of the ultimate high-tech genetic mapping or physical mapping, down to the level of the sequence, what you're looking at here is, if I go back for a second, you can't even guess what this is, right, from the jump in scale. If we can go back, there's a park. The blue square identifies a park. In the center of that park, there is an individual lying on the lawn, and this is his hand seen under a magnifying glass. So that's the level of the sequence. And I think you can see we have a problem, because we're going to need both conceptual and methodological methods of progressing through all these orders of magnitude without getting lost. Okay, now we want to make maps. We want to understand and be able to study, characterize the structure of the genome. And I'm going to be talking about 4 types of maps. And Eric has really introduced these for me, which makes this very simple. Physical maps are those in which distances are measured along the DNA molecule. They correspond to actual, tangible physical structures. The ultimate physical map is the sequence, which is just the highest resolution possible. It describes every base. And it's a lot of work to get it, and it's rather cumbersome to work with it. It's almost impossible to work with the sequence by hand, I feel. A much more limited but very similar physical map is a restriction map, which instead of giving you the entire sequence, provides just the distances between certain subsequences, between as it happens, the sequences recognized and cut by particular enzymes. The reason why this kind of map is so convenient is that by simply measuring the size of a fragment produced, let's say this one, by a set of enzymes, you know the distance between the sites. And I will spend a fair amount of time showing you the current state of progress in constructing restriction maps, because that's what we do. Now, both the sequence and the restriction map are really abstract quantities. They describe the genome, but they're going to exist as we get more information only in computer memories. They're really not going to, they're not something that you actually possess. On the other hand, an ordered library, like a cosmid map or an ordered map of yeast artificial chromosome clones, is actual physical possession of the genome. It's a much more powerful tool because you own the genome at that point. You have all the materials. And such a map consists of let's say, 50 or 100,000 vials of frozen samples in a set of freezers. And one of the problems, which I'll touch on later on, it's by no means a simple problem, is how do you actually handle such a map once you have it? How do you handle and distribute 50,000 samples without anybody making mistakes with them? But in any event, all of these maps are physical maps. And they must be contrasted in a very critical way with a typical genetic map, which for humans is a linkage map based predominantly on recombination frequency between markers. Now, Eric Lander has described the linkage map and already illustrated for you what one of the problems is that makes it not so straightforward to pass between a linkage map and any kind of physical map. We know that the order of markers, the relative order of markers in any genetic map and a corresponding physical map must be the same. But the relative distances between pairs of markers can vary alarmingly when physical maps and genetic maps are compared. It turns out that in bacteria, where we now have some genetic and physical maps, and in yeast, where we also have these maps, the discordance in relative distances between the two, physical and genetic maps, is not very great. But we know, especially in the mouse, where there's some very detailed mapping data available in the major histocompatibility complex, we know that there are tremendous variances in what distance means when you compare physical and genetic map, variances of a factor of 100. So that can be extremely confusing, and it's very important to keep in mind. But the major thing that distinguishes the physical maps from a genetic map is their much higher resolution, at least in principle. It's very difficult to talk about a genetic map with a resolution higher than 1 million base pairs. And today's map has a resolution closer to 10 million base pairs. It's very easy to talk about physical maps with resolutions of 100 or 200,000 base pairs. The ultimate resolution, of course, is a single base pair in the sequence. All right, now, these maps are maps of chromosomes, and we're interested in some of the following problems as mapping problems. We have made a map of the E. coli genome, which is 5 million base pairs in size, and a map of the S. pombe genome. S. pombe is a simple yeast. It has 3 chromosomes. Altogether, they total 15 million base pairs, and that map is now complete. We are in the process of making a physical map of human chromosome 21, and I will talk about that in some considerable detail in a bit. The largest human chromosome is 5 times bigger, chromosome 1. That's 250 million base pairs, and the whole human genome, here again, is 3 billion base pairs. Now, each of these individual chromosomes, let's say chromosome 21, consists, as far as we know, of a single linear, for human anyway, and bacteria sometimes circular, but for human single linear DNA molecule that stretches the entire length of the chromosome. And if we look at what that actually looks like as a molecule, it is a very complicated thing. This is actually a picture from Uli Lemle, and it shows a human, or maybe a mouse, but in any event, a mammalian metaphase chromosome viewed under the electron microscope. The cross structure here that I'm trying to outline is a sort of residual protein precipitate that represents the scaffold of the metaphase chromosome. And almost the entire rest of the slide is covered by this gray thing, which if you have good eyes, you may be able to resolve as a DNA molecule. If you look over at this edge where the pointer is, you can see the molecules sort of snake in and out. That is, what you're actually looking at here is about 50 million base pairs of DNA as a single molecule. And this is the kind of molecule we're actually trying to work with. Now, we can't quite manage that. When we started trying to work with large DNA molecules, this was the largest DNA that anybody could conveniently handle. It's the single DNA molecule of bacteriophage lambda. It's about 50,000 base pairs in size. We know its entire sequence. We know everything about it, just about. And between this molecule and an intact chromosome is a factor of 1,000 in size. And what I'm going to show you over the next few minutes is that we've developed methods that now allow us to work with DNAs that are range in size from this DNA, 50,000 base pairs, to DNAs that are larger than 5 million base pairs. So we've come most of the way towards being able to handle even intact DNAs from entire chromosomes. Now, in order to work with these large DNAs, we've really had to invent a whole series of new techniques, because the large DNA molecules are so fragile that you can't handle them in by ordinary solution techniques. DNA molecules are subject to what's called shear breakage, just mechanical breakage, and this effect scales as the square of the size. And above a certain size, the molecules are just always broken into pieces, if there's any possibility for convection or mechanical stress. And to get around this problem, what we've had to do is to move all DNA biochemistry into the solid phase. This, for example, shows the way in which we make our DNA samples. The actual slide illustrates this for bacterial DNA, and in fact, for human DNA, it's even simpler. What we do is take a suspension of healthy growing cells, although we've now recently learned that this can be done with frozen tissue, which is surprising but very pleasing. And we mix it with molten agarose and embed the combination into a mold, pour the mixture into a mold. And this results ultimately in embedding the cells in a matrix of agarose. And then we diffuse into the agarose whatever enzymes, detergents, salts, chemicals are required to successively eliminate all of the constituents of the cells except for the DNA molecules. So, for example, in the case of bacteria, we would diffuse in first lysozyme to cut the cell wall off. That would produce protoplasts in the agarose in situ. We would then lyse these protoplasts with high salt and detergent, and then diffuse in various proteinases and ribonucleases to digest proteins, digest RNA, until finally what's left is intact chromosomal DNA molecules sitting in the holes where the cells used to be. Now, this turns out to be an exceptionally convenient and effective procedure. And the reason why it works is because proteins, detergents, salts are all much, much smaller than the 100 nanometer pores in a typical agarose gel. So they diffuse readily in and out of the gel. But chromosomal DNA molecules are much, much larger than the pores in the gel. So under ordinary circumstances, they can't diffuse at all through the agarose, and they sit there entrapped. One of the advantages of this is that the procedure then has 100% yield. You recover all of the DNA that you started with. Another advantage of this in principle is that absolutely no equipment is required. The entire preparative procedure from beginning to end just consists of bathing these little pieces of agarose in different solutions successively, one after the next. So in principle, this sort of procedure would readily be automatable, but that's something that no one's done in practice yet. Now, in most of the cases I'm going to show you, these intact chromosomal DNA molecules are far too large to work with directly. And so what we do is we then diffuse restriction enzymes into the gel, allow these enzymes to, let's see, my pointer refuses to go further to the right. I wonder why that is. Okay. You can see, however, from what's written, that we diffuse restriction enzymes directly into the gel. They cut the DNA molecules into fragments in situ. We then remove the restriction enzymes, and then we're ready to actually try to fractionate these large DNA pieces in order to basically resolve them and analyze them. Now, I'm not going to talk about this in any detail. I just want to show you how we cut these molecules into pieces. It turns out that most restriction enzymes that cut DNA at specific sequences recognize sequences that are 4 bases long or 6. And except in rare cases, those sites are far too frequent in genomes to be usable to generate the kinds of large fragments we need for effective physical mapping. But there are 2 restriction enzymes, NOT1 and SFI1, which recognize 8-base sites. NOT1, the site is particularly rare in the human genome and is a very, very effective enzyme for us. SFI1 has a problem in that it's an enzyme which recognizes a sequence that is interrupted. And such enzymes usually have some kinetic specificity for the intervening bases, and therefore it's very difficult to get these enzymes to actually digest to completion. So this enzyme is useful in principle, but in practice is not so pleasant to work with. And what we're able to do with these enzymes is take a genome, cut it up into a finite number of large fragments, and then we've developed techniques for fractionating these very large DNA pieces. Now, those techniques basically consist of making the DNA molecules continually change direction as they move through a gel matrix in a strong electric field. It turns out that if you apply an electric field to large DNA molecules, they rapidly orient parallel to the field. Once they do so, then they move through the gel matrix at exactly the same velocity, regardless of their size. And so it's impossible to separate DNA molecules in an electric field if they're going straight, once they get above 50,000 base pairs in size. But the trick that we developed 5 years ago was to recognize that the molecules change directions at different speeds, so that the small molecule can reorient in the gel very quickly. A large molecule takes a very long time to reorient. And in fact, as far as we know, the effect scales linearly with size, everywhere from 50,000 base pairs up to more than 10 million base pairs. And so by simply tuning the time scale on which the molecules are asked to reorient, we can basically spread out our separation range over any size scale desired. And I'll show you an example here for the E. coli genome. First, let's start with this lane on the left, which are size markers. That's lambda monomer, which is 50,000 base pairs in size. This is a dimer of lambda, which is 100,000 base pairs in size, a trimer, which is 150,000 base pairs, and so on. So this separation actually covers the size range from about 20,000 base pair fragments up to about 400,000 base pair fragments. What you're looking at in all of the center lanes is the E. coli genome that's been cut with a restriction enzyme, and then the fragments have been separated. And you'll notice that most of the lanes contain a very large number of relatively small fragments. A typical restriction enzyme will cut the coli genome into about 1,000 fragments, averaging maybe 20 or 30,000 base pairs in size. These are far too numerous and far too small to really be useful for any kind of rapid mapping. It would be like putting together a jigsaw puzzle with 1,000 pieces, except you can't see any of the pieces. But the two 8-base specific enzymes generate a much smaller number of very large fragments. In this lane is the digest with NOT1, and here is the digest with SFI1. NOT1 actually cuts the coli genome into 22 fragments, and we're able to fractionate all of these by length in one pass. SFI1 cuts the genome into 27 fragments, and we can basically resolve most of those as well. Now, putting together a restriction map, making the simplest physical map, means two things. It means identifying each of these fragments, where in the genome do they come from, and then proving the actual order, proving, let's say, that this fragment in the actual genome might be next to that one. And the types of methods that we use to do this are summarized here. But before I do that, let me just contrast for you the simple case of E. coli with a much more complicated case of the human genome. Here I have E. coli fragments separated again, but now I'm using a much longer time scale, and so most of the E. coli fragments have run very rapidly and are seen near the bottom of the gel. This lane shows lambda DNA, monomer, dimer, trimer, higher oligomers, and you can see how much finer the ladder of size standards is. So in fact, this gel actually is separating fragments up to about 1 million base pairs in size. Now with E. coli, we see a small number, 22 fragments. We can resolve them all. With the human genome, we estimate that the same enzyme, NOT1, will give about 3,000 to 6,000 fragments. And under those circumstances, you would not expect to see anything except a featureless smear, and simple statistics would predict that it would be roughly a Gaussian distribution of fragments centered around some mean value. And I think you can see very clearly that the human genome is not behaving in a simple statistical way at all. And that's the immediate surprise that we got when we started to do these experiments. First of all, the overall distribution is two-phase. There's a lot of very large fragments, averaging anywhere from 1 to 10 million base pairs in size. There's a lot of what I call small fragments, ranging from basically 500,000 base pairs down to less than 100,000 base pairs. Very little material, actually, between about 500,000 base pairs and 1 or 2 million base pairs. Very surprising. There's something odd about the statistical distribution of the sites that are recognized by this enzyme. The second thing which is surprising, and we'll come back to it a little bit later, is that instead of seeing a featureless smear, we see bands. It's as though there are certain preferred distances for these supposedly arbitrary sites recognized by this particular enzyme. All right. In any event, what we have to do in the case of E. coli, or in the case of the human genome, to make physical maps, is to identify fragments and determine their order. And since we can't see most of the fragments, we have to use radiolabeled probes in order to sort out individual fragments for us. And this slide illustrates the 3 ways that are currently being used to try to put together restriction maps of entire chromosomes. The most elementary is simply called the southern blot. You separate large fragments, you transfer them from a gel to some kind of membrane filter, and then by using hybridization, you analyze individual fragments that happen to correspond to any cloned ordinary sized piece of DNA that you have available. So for instance, if, as in the case of E. coli, or certain regions of the human genome, you have a detailed genetic map, then you can take individual genes cloned from a particular region, make them radiolabeled, and use them to, the genes in the case of E. coli might be 1,000 base pairs in size. And you can use that small piece of DNA to identify the corresponding large piece of DNA on which it sits. And so you basically gain an amplification of a factor of 100 or more in size. Now, for example, if you know on the genetic map that a certain cloned gene is near another one, and one of these identifies fragment X, and one of these identifies fragment B, then you can begin to suppose that fragments X and B are located near each other in the genome. The difficulty with this approach is, first, that it's very slow. Each experiment identifies only a single fragment. Second problem is that it's not rigorous. Just because your genetic map says that X and B are close doesn't mean that there isn't some other fragment between them. And furthermore, since the genetic map and the physical map don't have to correspond in terms of relative distances, there's a possibility that you could be totally confused. So to get around this problem, we've come up with two other methods which are much more powerful. The first of these uses a specialized types of DNA probes called linking probes. Here, you, by various tricks, isolate a small piece of DNA that happens to contain the same recognition site that was used by the enzyme in order to cut the whole genome into large fragments. And since that same little site is on this probe, then when used in hybridization, the probe must detect, because it must correspond to, both fragment C and fragment X. The method's twice as fast because you identify two fragments with each probe. But much more important, it's far more robust because it actually proves the two fragments are next to each other. And in principle, there's enough information available in a complete set of these probes to piece together the entire puzzle, two fragments at a time. The major difficulty is it's still very slow. And to try to gain more speed, we've spent a lot of energy developing the ability to do partial digests. Here, instead of allowing an enzyme to go to completion, we allow it to just cut the various sites statistically and generate a huge set of fragments consisting of all unique fragments, all adjacent pairs of fragments, all adjacent triples, and so on. And then by using a radiolabeled probe, we're able to identify fragments that correspond to a parent band, the sum of its size with neighboring bands, and so on. In principle, this is very powerful because the information tells you about adjacency, and also because it identifies large numbers of bands at once. The difficulty is that the data is a convolution of fragments reading to the left and reading to the right, and it's not always easy to sort that out. There are special tricks that can be used to help do it, but a general solution actually, I think, requires some fairly good data and fairly exacting analysis, and I'll show you some examples of that later on. Okay, well, using this type of technique, Cassandra Smith was able to make a complete restriction map of the E. coli genome, and I show that to you here. The actual physical map is drawn as the relatively thick lines extending around the outside. There are actually 21 fragments in the parent genome, and the 22nd was an episome, an F+ plasmid, about a 100,000 base pair plasmid in this actually E. coli wild-type strain. That's the physical map, and drawn, basically distorted in order to fit it around the outside as a thin line is the genetic map. And you can see that in the case of E. coli, the distortion needed to fit the genetic map to the physical map is very slight. This represents the worst region right here, where there's about a factor of 2 change in the metric in comparing these two maps. That's the worst case, at least at our current level of resolution. Now, it took about 1.5 years for a team of 3 people to put this map together. But Cassandra estimates that to do it again, using the most powerful methods currently available in the lab, would take only a month. So one of the major things I want to leave you with is that physical mapping has become very easy and sort of effective way to learn about the structure of chromosomes. And I think the best way I can convince you of that, I'm not going to show any details, but the entire physical map of S. pombe, a genome 3 times the size of E. coli, was put together by a single first-year graduate student as a rotation project working for 1 year. And since first-year graduate students at Columbia have to take courses during the daytime, all the lab work was done at night. This guy successfully completed a map of the S. pombe genome. These projects, however, are well, and just to remind you how far we've come, that dot drawn to scale, sitting right below the pointer, is a PBR 322, an ordinary 4,000 base pair DNA plasmid. So we can make a map of the E. coli genome today with not much more effort than was required to make a map of that tiny dot. Of course, the map's less detailed. Well, the same kinds of things are directly applicable to the human genome. And what I want to do in the next few minutes is just give you a feeling for our current state of progress in trying to make a map of the smallest human chromosome, chromosome 21. We're learning rapidly that this is actually a far more difficult task. This is 10 times the size of E. coli, 3 times the size of S. pombe. But part of the problem is that the quality of the available resources in terms of clonal cell lines, in terms of well-characterized probes, in terms of reliable sort of genetic information to start with, the quality of all the input is far worse. So the problem is much harder than the size scale would indicate. We chose chromosome 21 for 2 reasons. It is the smallest human chromosome, as far as anybody knows, and essentially one of the 2 most densely mapped. That is, there's more material available from this chromosome if you scale it according to size than I think almost anything else except the X chromosome. All right, so the first thing that you actually do is you collect from everybody who's willing to give it to you every piece of DNA that has been cloned and characterized and identified from this chromosome. And when we started the work, there were some, I guess 100 pieces of DNA that we could imagine that we were interested in, and we were able to obtain most of those. Unfortunately, a significant fraction of them turned out to be unreliable. That is, the pieces of DNA did not match up with their supposed credentials. That is a real problem in human molecular biology, much more serious for some reason than in E. coli or yeast molecular biology. In any event, we have these large probes, and they have been large set of probes, and they have been regionally assigned to this chromosome by basically mostly somatic cell genetic methods. We also have a smaller number of probes that have been genetically mapped onto chromosome 21 by the methods that Eric Lander described. But most of these probes are assigned by either in situ hybridization or by using somatic cell hybrids. And the precision of that assignment is really terrible, frankly. If you can see, these brackets indicate the known position of various probes, and you can see that the brackets in many cases cover relatively wide ranges of the chromosome. The way in which this is done is shown here, for example, for somatic cell genetic methods. You take a cell with just a single human chromosome, or if you're really lucky, you take cells that have just portions of human chromosomes. They may have just the upper 2/3 from a translocation, or they may be missing something in the middle from an interstitial deletion, or they may be circular and be missing the ends. By the way, my pointer is degrading from the right-hand side. So this is now the first. Oh, no, thank you. All right, I just got some of it back again. I think, no, I didn't. OK, the pointer is dying, friends. All right, in any event, the pattern of hybridization of any given probe to these various cell lines lets you, by using simple binary logic, sort out where the probe came from. The difficulty is that your information is only as good as your cytogenetic observation of where these anomalous chromosomes actually differ. And that's a severe limitation. All right. Now, the next thing, having collected a bunch of probes, having characterized them or accepted other people's characterization of them, we wanted to get some measure of how complicated the physical map of chromosome 21 was going to be. And to do that, we took advantage of these hybrid cells in the following way. If we start with a hybrid cell containing just a single human chromosome, and we cut it with a restriction enzyme, and we separate all of the pieces using a pulse field electrophoresis, then if we sort of stain the DNA with just a colorimetric stain, of course, we'll see all 6,000 fragments that correspond to both the rodent genome and the human chromosome. But if we hybridize that gel with just human DNA or a highly repeated human sequence like the ALU sequence, it really makes no difference, then we will detect just those large DNA fragments that correspond to human DNA that happen to have highly repeated sequences on it. And in that case, we basically can just see that single chromosome. And the background from the rodent cell is absolutely 0. And that's shown here in 2 examples. Again, we're looking at a separation range that extends from about 50,000 base pairs up to 1 million base pairs. This lane on the left is human chromosome 21 living in a mouse cell. And this lane to the right of it is human chromosome 21 living in a Chinese hamster cell. These 2 lanes immediately adjacent are the control mouse and hamster cell. And you can see they're absolutely blank, because the probe that we have is totally specific for human DNA. Now, we're able, by looking at these bands, then, to form an overview, an aerial view, of that entire chromosome. Using these separation conditions, we see about 15 or 18 bands, and using a longer time scale, we can resolve those larger fragments. And altogether, we see almost 30 bands from this chromosome. And they amount to about 30 million base pairs of DNA. So we're seeing, with a single probe in a single experiment or set of experiments, something like 2/3 of the known chromosome. Now, that was a little surprising, frankly. We expected to see all of the chromosome, because the sequence that we're using to probe this chromosome occurs 500,000 times in the human genome. And that means that roughly every 6,000 base pairs, the sequence should be there. And since our average fragment size is hundreds of thousands of base pairs, the number of those fragments that should not contain a single copy of this repeated sequence should be vanishingly small. But it turns out, thanks to some work that's recently been done by Bob Moises at Los Alamos, it turns out that, surprisingly, on this particular chromosome, there's a large patch that apparently is quite deficient in this highly repeated ALU sequence. And so we're not able to see probably about 1/3 of the chromosome for that reason. All right. Now, I want to show you one more thing here before we leave this, because it's a source of a remarkable problem and also a remarkable opportunity, which I'll come back to once or twice as I continue to talk. These two chromosome 21s came from different individuals. And they're different because of natural polymorphism in the human genome. Furthermore, this one's living in a mouse, this one's living in a hamster. There's an endogenous methylation pattern which blocks some of these restriction sites, blocks some of the enzyme cutting sites, and that's species-specific, and it's also cell-type specific. And so for both those reasons, we see quite a different banding pattern in the two samples. For example, that band is missing in that cell line. This one is missing. And there are lots of other differences. And to make a long story short, from our aerial perspective of sort of megabase scale, we see enormous polymorphism in many regions of the human genome. And that is a source of great frustration if you're trying to construct maps, because it means that your map ultimately either is not generally applicable or represents a synthesis of the maps of many individuals. But I'll try to convince you it's actually an advantage, a tremendous advantage, if you start to think about it properly, because that polymorphism provides tremendous information. OK, now, I want to just summarize for you, if you can zoom back, where we are today in trying to map chromosome 21. This slide is somewhat out of date, but it still gives you a fair picture. We used about 30 of the 50 well-characterized probes that we had at this particular time. Most of these were able to be successfully used. That is, they actually hybridized to something that we could detect and get some data. And roughly, we can now find something like 1/3 of the ALU fragments. We know where they correspond to on the chromosome. And altogether, by using the DNA detected in this way and other DNA that we know is on the chromosome, we can actually account for something like 38 million base pairs of DNA, but all of it in disconnected pieces at the moment. So I estimate that we're probably about halfway along in trying to make a physical map of this chromosome. But that's very misleading, because anyone who's done mapping projects knows that it's very easy to start a map and very hard to finish one. You spend roughly 90% of your effort uncovering the final 10% of the map. OK, now mapping is very boring. To those who do it, to those who listen to it, it's terrible to have to show maps, because unless you actually want to use them, who cares? What you hope as you construct a map of the human genome is that patterns will emerge that are interesting, that provide some clue to function or organization. And fortunately for us, I think we're clearly beginning to see that there are some rather striking patterns, certainly patterns we didn't expect, and patterns that will make the whole task not only interesting, but much more useful. And one of the first clues that there's something amusing going on here comes from the sizes, just the sizes, of the fragments generated by an enzyme like NOT1. And this shows a summary of fragment size for 3 of the particular mapping projects we've been involved in: chromosome 21, the major histocompatibility complex, and Huntington's disease, which is the terminal 4 million base pairs on chromosome 4. And what I show is the sizes of the fragments that we've seen now, different fragments identified with different probes in each of the 3 mapping projects. And you can see that there's something very strange going on here. With Huntington's disease, in that region of the genome where we now have 14 distinct, isolated clone fragments, 10 of the 14 detect NOT1 fragments less than 100,000 base pairs in size. Only 4 of them detect fragments in the 0.1 to 0.5 megabase range, and there are no larger fragments seen from that region. What that means in terms of mapping is this region is an utter disaster, because nothing we can do shows it to us all at once. We're sort of really just digging it through the ditches, a worm's eye view of that region. On the other hand, chromosome 21, fortunately for us, the recognition sites for this particular enzyme seem to be terribly rare across the whole chromosome. More than half of the DNA probes that we've used detect fragments that are larger than 1.2 million base pairs in size, and absolutely none. We haven't found a single case yet of a fragment less than 100,000 base pairs in size. So there's something interesting going on here. And I think the first reaction that certainly anybody with a mathematical background will have is, well, this is nonsense. I mean, it says it's just one particular enzyme. These are just statistical fluctuations, So try another enzyme. And what you find, if you try another enzyme, is a correlation in fragment size between 2 enzymes which have, at least on the surface, totally unrelated recognition sequences. This shows the size. This is actually data from chromosome 21. And it shows the size fragment detected by the enzyme MLU1 on the top, or NOT1 on the side. And you can see the correlation is absolutely fantastic. Whenever MLU1 detects a small piece from the genome, so does NOT1. And whenever MLU1 detects a very large piece from chromosome 21, so does NOT1. All right? So it's not just a trivial statistical artifact. What's going on here? It turns out, fortunately for us, that all of these enzymes recognize special kinds of sequences which appear at least to have special, functionally interesting locations in the human genome. And the first evidence for this comes from just looking at the sequence. NOT1 has 2 CPGs in its recognition sequence. The sequence is GCG, GCC, GC. MLU1, here it is only a 6-base specific enzyme. It generates enormous fragments from the human genome. Its site is very rare. And it has 2 CGs in its recognition sequence, A, CG, CGT. Similarly, other enzymes, in fact, all enzymes that are known thus far, that generate very large fragments from the human genome, with the single exception of SFI1. All the others have 1 or usually 2 CPGs in their recognition sequence. This is not, in fact, surprising. We know, it's been known for quite a while, that the sequence CPG is extremely rare in mammalian genomes. The human genome, other mammalian genomes, are roughly 40% G plus C, 60% A plus T. So you'd predict, by simple binomial statistics, that the sequence CPG ought to occur at the level, I think, of something like 4%. But it actually occurs 5 times less frequently than would be expected from any simple binomial statistics. And the reason is that the sequence is being systematically deleted from the human genome by mutation. The reason why it's being lost is that most, roughly 80%, of the CPGs in the entire human and other mammalian genomes are methylated. It is the primary DNA modification site in eukaryotic genomes, higher eukaryotic genomes. And so most CPG is methyl CPG. And when methyl C is deaminated, and deamination is the most frequent mutational event that occurs in DNA. When methyl C is deaminated, it becomes the base T. When ordinary C is deaminated, it becomes the base U. Since U does not exist in DNA, a specialized repair system has evolved to recognize that if there's a U, it has to be wrong, and you delete it. And so you restore the mutation back to the original correct genotype. But when you have a methylated C, when it's deaminated, it becomes T. That's a natural occurring base in DNA. The repair system cannot tell which strand had the mutation, and so 50% of those cases revert with a resulting mutation. And that means the sequence will be lost wherever it is not necessary to preserve function. And where we are today, after however many millions of years of human evolution, is we've lost something like 80% of that CPG. Okay. Now it turns out that many of the remaining CPGs in our genome, the 20% that are not methylated, the ones that will not be lost because they don't have this problem, they occur, for reasons that aren't clear, in special places in the genome. They are clustered together. They occur in GC-rich sites that average about 1,000 base pairs in size. And that tend to be next to housekeeping genes. That is, genes that are expressed in all tissues. So when we cut a mammalian genome with any one of these enzymes that recognizes CpG and is blocked by methylation, the only place that enzyme can cut is in one of these islands of CG-rich sites, and that is located next to a housekeeping gene. So when we make what could have been a very, very boring physical restriction map of the human genome, what we're actually making, more or less, at the same time, is a map of the location of a certain class of genes. And so when we start to see patterns like clustering of these cutting sites on the tip of chromosome 4, what that says is that region of the genome is full of housekeeping genes. And then it becomes more reasonable. You can imagine how a region might evolve and stay localized. And it says that chromosome 21 has very few housekeeping genes for whatever reason. Right? So we're going to see, I think, then that there are lots of patterns. I think this is probably only the first one that will actually emerge. Now, I've talked thus far totally about the top-down approach to mapping, and because it's what I do at the moment. But I want to mention that there is an alternative approach, which is equally powerful and which we're beginning to get interested in ourselves, and that's bottom-up. In top-down, you make a restriction map. In bottom-up, and Eric Landon referred to this already very briefly, you start with small fragments, and you look for overlapping patterns on these fragments that allows you to link them up and determine the order. And so you make contiguous assemblies of these fragments. The advantage here is that if these can be cloned fragments, and so you own these pieces. And in fact, what's done is to fingerprint these. And Eric showed one example. I'll just describe it more simply here. It turns out that if you have a clone of 50 or 100,000 base pairs in size, it's possible to cut that clone with one restriction enzyme, label at every place where you've cut with a second, and generate then discrete but radiolabeled pieces. And so each of these little chunks of the genome will have a length, 126 nucleotides, 157 nucleotides. And if you choose the right enzymes, and then these numbers, these fragment lengths, which you will see by separating the fragments on the same kind of gel electrophoresis that you would sequence, these numbers are an absolutely accurate diagnostic measure of this region of the genome. And if you have two fragments of the genome that overlap, then they'll have, for example, you might see that both of them have produce little pieces, 84, 37, 196, and that tells you that, in fact, they're contiguous clones. And so you can piece together an overlapping library of fragments by such fingerprinting. Well, this method was basically pioneered by Coulson and Sulston, working with Sidney Brenner at the MRC, and it was designed at a time when the largest pieces of DNA one could clone were only 20,000 base pairs in size. And where in order to do that, one had to clone basically random chunks of DNA. And so the ends of each clone were undefined. Right? But today, because of yeast artificial chromosomes, we have potentially a much more powerful method, which I predict will essentially take over this link up in basically no time. Because since we can clone now pieces of DNA that are hundreds of thousands of base pairs long and have absolutely specified ends, in this case it's shown for NOT1 enzyme cut ends, then in order to link up this piece with that piece, all we need is one clone, a linking clone, which contains this same particular NOT1 site, because in hybridization, just as with gel fragments, this clone will hybridize just to two specific large yeast artificial chromosomes, and will tell you they have to be adjacent. So once again, with a relatively small number of clones and a relatively small number of these linking clones, you could piece together the order of the entire genome. And I think that's the kind of activity that's now ongoing in a number of different laboratories. I don't want to minimize the technical problems. There still is one major problem to be resolved here. The current ability to clone DNA in yeast artificial chromosomes is peaked around 200,000 base pair sizes. It's very difficult to push it with any kind of efficiency at the moment above about 400,000 base pairs in size. The average NOT1 fragment among the smaller set of this biphasic distribution is a few 100,000 base pairs in size. That's fine. But half the genome is in fragments larger than a few million base pairs in size and are not clonable at all by the present technology. All right. Now let's go back to the basic theme that you heard in Eric's talk. Why do we do this? Well, ultimately, we do this to find genes, and genes we want to study. And the typical situation which exists today, and will exist even in the future, is a genetic map with a marker that is linked by the methods that Eric described to a gene that you're interested in, but is millions of base pairs away from that gene. With today's genetic map, this distance is 10 million base pairs on the average. With the genetic map that we project, if we're lucky, 5 years from now, with 5 times the resolution, this distance might only be 1 or 2 million base pairs, but it's still a long distance. And what we need to do is to find ways to go from any marker that we have to one much closer to the gene. Now, when we have an actual linked library from the human genome, when we actually own all the DNA from this region, then that will simply require going to your freezer, picking out a clone that lies the appropriate distance away from the one the marker's on, and saying, I'm probably near that on this clone, on this 100,000 base pairs of DNA, is probably the gene I'm interested in. Now, there are 2 problems. One is trivial, and that is that you don't know the relative orientation, so that 50% of the time you'll have to look to the left when you should look to the right. Say you take 2. All right, that's not so bad. The more serious problem, which I keep trying to touch, is that the stretchiness of the human genetic map. That is, you don't really know how to translate the genetic distance to a physical distance, so you could overshoot or undershoot. But nevertheless, it's going to be much more efficient to jump a few million base pairs at a time than it is to jump only 10,000 base pairs at a time, which was what was possible until just very recently. Now, even without possessing all the DNA in the region, it's possible today to start with that marker and jump. And the reason is the following. The sites cut by most of these enzymes that yield big fragments turn out to be extremely polymorphic. The reason is that the sequence CpG is, in fact, a major site of human polymorphism. For the reason that I've already told you. It's a highly mutagenic site. And so it's natural. It's a high mutation rate for that site in the genome. It's natural that sequence is going to be polymorphic. These enzymes recognize 2 CpGs. So the polymorphism frequency is the square of what you'd ordinarily see. And what that means in practice is it's not difficult to find polymorphisms using these particular enzymes. And so in many cases, in fact, just the restriction map of this region will yield another polymorphic site, which is likely to be closer to the gene that you're interested in. Okay. Now, Eric already mentioned, and I'll sort of reiterate, how do you know a gene when you found it? How can you tell if you've got the right thing? And the answer which you already gave is we don't know how to solve this problem yet, unless we're lucky. We have found a number of genes in the last couple of years using these basically reverse genetic approaches. How has that worked? Well, in a couple of cases, we were able to find the genes because we had mutant alleles that were deletions. Well, in a couple of cases, we were able to find the genes because we had mutant alleles that were deletions. That's certainly the case in Duchenne dystrophy, where a large fraction of the mutations that you see are big deletions. So if you have a big deletion, the moment you have a map of the region, what you see is a fragment is too small. That more or less puts you right into the gene. All right? So in those cases, the problem is trivial and instantly solved. Okay? But in the majority of cases, it's not going to be that way. The majority of the mutations are probably going to be point mutants. I mean, we know that from the very limited studies that currently exist on mutagenesis in other organisms. All right, with a point mutant, let me just show you the depressing statistics. The genetic map will isolate you, localize you, to a region today of 10 million base pairs. 5 years from now, 1 million base pairs. If you decide this is the most interesting thing in the world and you plow into it tremendous resources and effort, you might be able to narrow it down. The most recklessly optimistic people say 100,000 base pairs. That would require an effort, the cost of which is almost inconceivable. But it's not impossible for a frequent genetic disease anyway. All right. Now, in 1 million base pairs, let's stick to the middle of the road, the realizable not today but tomorrow. In 1 million base pairs, since an average human gene is of the order of 30,000 base pairs, there'll be 30 genes. And so you have to find which one of 30 is it. If you have the sequence of the whole region, you can undoubtedly identify the 30 genes and know what they are. But which one is causing the disease? Well, you have two chances. If it's a disease where there's some likelihood that the expression will be altered in a very tissue-specific way, then it's possible to look at what tissues these genes are expressed in and maybe narrow down quite substantially which ones really should be the candidates. I mean, if it's a kidney disease, it's unlikely that it's going to be a gene that's expressed only in the brain. Okay? You could get fooled, right? But in most cases, I think you'll help yourself a lot. Now, unfortunately, let's take the case of Huntington's. The Huntington's disease gene is in a region where almost everything seems to be housekeeping genes, and there are probably 100 of them. So in that case, it's expressed in all tissues. It's not going to help you. All right. So in the worst case, and this is the worst case, the only thing you can do is sequence. If you can narrow it down, and here it's shown for the recklessly optimistic example of 100,000 base pairs, because the polymorphism frequency in the human genome is 0.1%. 100,000 base pairs will have 100 polymorphisms. And the only scheme I know to tell which of these is actually causing the disease, if it's a single point mutant, is to take a collection of individuals who are all affected and sequence the DNA from all of them in this region. Take a set of individuals who are all unaffected, sequence all of them, and look for a pattern of polymorphism which is absolutely co-inherited with the disease. And that will not necessarily narrow it down because of a phenomenon called linkage disequilibrium, but it should at least get you as close as anything can get you. All right? And if, in fact, you argue, I estimate that about 10 individuals is, for this particular case, is what it would take. And if you could only localize to within 1 million base pairs, then you'd probably need more like 15 individuals rather than 10 individuals. Well, that is not ridiculous, actually. It's not ridiculous in terms of the technology for automated DNA sequencing, which is now just beginning to exist. But at today's dollars, it's still rather expensive. All right. What I want to do now is to turn to what you're probably more interested in, which is not what we can do, but what you can do. And so I want to talk about computing needs as I see them, both present and future. And I'm going to divide this into 4 categories. Actually managing the kind of mapping data that I briefly sketched for you, analyzing that mapping data, managing sequencing data, and analyzing sequencing data. And let me sort of give you an apology in advance. This is a user's view of this, and not a computer scientist's view of this. And I'm probably going to insult a few of you by arguing that we need something which you know we already have. Okay? But in that case, it's evidence for a communication gap, because if you already have it, but I don't know about it, and I need to use it, then you better let me know. And also, you have to, although I actually am not totally computer ignorant, the average molecular biologist that you need to address is, for all practical purposes, maybe they can use a word processor. So you have to get it down to the Macintosh level. I think that's the bottom line of what I can say. Nothing else is going to work for your user community. And I think there are a lot of very interesting problems. Okay, now maps, by which I mean really genetic maps, physical maps, restriction maps, cosmid maps, all kinds of maps, but not sequence. The data is actually read, interpreted, and input into computer databases by hand. Okay? As far as I know, none of this, there's no automated data entry available today for any of these procedures. The updating is periodic. That is, these databases are not kept up to date instantaneously, which it makes sense if you have to put the data in by hand. And in the case of the human genome genetic map, the updating was biannual until, I guess, this summer, when in a revolutionary spirit, it was decided to make the updating annual. I mean, and I think that's a very bad scene. And one needs to get some modern database management into the field of human genetics, which will probably have to be done in part, or it is being done, really, over the kicking and screaming of some of the people in the field, but nevertheless, it has to be done. Now, to make things worse, different types of databases are largely unlinked. That is, there isn't a database of database. There isn't any kind of simple way that lets you pass from sequence data to genetic map data to physical data. And to show you just how bad the current situation is, in the latest Human Gene Mapping workshop, a number of us who were making restriction maps, in fact, roughly 15% of all the posters presented at the meeting had macro restriction maps, which was absolutely amazing considering that a year before there was no such map in existence in the genome. Suddenly, 60 million base pairs of the human genome had been restriction mapped. And so we raised the question, can we please incorporate all of this restriction mapping data into the genetic map? And the answer was no, can't do it, don't know how. Come back next year. And so the maps are totally unlinked. And it's also, it's not a trivial problem, obviously, to link up maps which are drawn with different variable metrics and sort of are subject to totally different types of ambiguities. All right, now in terms of actually owning the genome, remember our goal certainly over the next few years is to own this genome, is to have it cloned. Sample storage is haphazard. There isn't any place you can go. There isn't a library where you can actually go, or a supermarket, and say, I want clone number 32831 or whatever. You can't do that, you know. And you write to somebody and say, can I please have it? And if they like you, they write back. If they get your letter, anyway. If they don't want to give it to you, they just pretend they didn't get your letter. And that happens a lot, as a matter of fact. Finally, and this is a very serious problem, actually, the sample nomenclature, what you call these things, is not standardized. And I think for those of you who are not very familiar with molecular biology, I should have to tell you that the way in which we name clones is a system in which there's a number which follows the initials of the person who cloned it. Now, if you think of trying to map the human genome, surely nothing is less relevant to the initials of the person who made this clone. How many people know that PSC 101 stands for Stanley Cohen and so on? I mean, it was fine when there were 5 people working in the field and they were all the pioneers of the field. But today, we can't go on with this. And I personally think that we're going to have a series of dictionaries constructed we need a systematic nomenclature for DNA clones. Someone has to think through. It's actually a very serious problem how to do this. Someone has to think through how to do that. And we also need to have various sets of dictionaries constructed. I mean, in our own case, just with mapping chromosome 21, we have 3 different nomenclatures, which we've been forced to inherit by other people who've sort of made large numbers of these clones. And we have our own dictionary to try to translate these, but it's a mess. All right, that's the current situation. In the future, I hope as soon as possible that these databases will be combined, that all the data handling will be automatic, the updating will be continuous, and that you will, by some kind of barcode method or whatever, be able to sort of dial up with your computer and say, please send me the following clones, and some automated robot will pick them out of the 50,000 clone storage bank and send them to you, hopefully without any errors. Today, something like 20 to 30% of all the clones we receive by today's system are incorrect, either because they've gotten scrambled by human error in the actual sending them to us or in the filing of them away, or because they were incorrectly characterized to begin with. That's the kind of error rate you simply cannot live with. Okay. And again, an argument for standard nomenclature. If anybody can think of a way to do this, I happen to sit on the International Nomenclature Commission for Biochemistry at the moment, so I might be able to get you a method adopted if there were a method. Map analysis. I think most comparisons at the level of genetic or restriction maps are today done manually. Construction of restriction maps is semi-automated. By that I mean there are programs that do it, but not many people use them. Macro restriction map construction is still manual because the methods used to link these maps up are different from those that have been where people have written programs thus far. The multi-point genetic mapping, as Eric told you, is already computerized, and ordered library construction is still developing as a technology. And so in terms of computational needs, I think they have to sort of follow in the footsteps of the actual biology technique development. In the future, I hope there are going to be robust methods for comparing maps, because as we generate more and more restriction map, it would be nice at the level of a restriction map to ask, is this restriction map similar to anything I've seen before in other regions of the human genome and other genomes and so on? Should be nice to search these maps for patterns, which is not easy because maps are very fuzzy kind of data. Certainly, we need to integrate data from different map types. And finally, in terms of macro restriction mapping, the kind of mapping I've talked about, we need to have automatic ways of analyzing the data. And I'll show you in a moment what I mean by some of that. This is the typical type of data we actually get when we try to map a region of the human genome by these large DNA techniques. What you're looking at is a partial digest which increases in stringency from left to right. So in the right-hand lane is a total digest. That's a parent band that a particular probe corresponds to. This happens to be the region near Huntington's disease, but it could be anything. And as you let the digest go not to completion, but only partly to completion, you see new bands appear that presumably represent doublets of adjacent bands, and then a much larger set of complex bands. And what we have to do at the present time to work with this data is basically to code all this information by hand. It would be very nice to be able to do it by image analysis. That's obviously a difficult problem when the image looks like this. And then we have to try to figure out, we have to infer or analyze this particular pattern of fragment sizes. And in doing so today, I should tell you that we do not have the ability to take into account information about the intensity of hybridization to particular bands. There's clearly information there. Why some bands are light and dark will have to do with cutting statistics, but we don't use it. We just use their sizes. And what we do, this shows actually the analysis of some of the data from that slide, is we had this particular probe here called, in lovely non-systematic way, HB1.4, and it identified a parent band and various neighboring bands. And if you have only a single probe, the left-right ambiguities are absolutely horrible. If you have more than one probe from the region, as we do in this particular case, then they start to sort out left-right ambiguities because the two probes together are more powerful than simply using each of them separately. Now, where we find ourselves today is in a very uncomfortable situation because we're accumulating more and more probes from this region, and it's simply impossible to update this analysis by hand. But yet nobody has actually taken this problem and devised the simple computer algorithms, although we've tried and we have some. But I mean, it's a problem which it turns out it's not a trivial problem to solve because in a real human cell, it's not haploid but diploid. And so the patterns that you're actually seeing are not necessarily coming from a single DNA molecule, but from a pair of DNA molecules which don't have to have the same pattern on them. And so the map is actually more complicated. All right. Now, I mentioned to you that the human genome was extremely polymorphic when viewed on the size scale of these large DNA fragments. And that is potentially a source of great frustration. But we believe, at least in principle, it's a source of great power. And in this slide, I attempt to show you why I'm saying that. Suppose we take 8 unrelated individuals and we study their restriction map together. Not just one, but we look at each of them. Okay? Then, because sites are polymorphic, because these sites are particularly polymorphic, then each particular restriction site will either be present or absent in each individual. And for the moment, let's imagine that these are just restriction site differences. So there's no other possibility except site present or site absent. It turns out that through the set of individuals, then the pattern of a particular polymorphism, who has it and who doesn't, can be coded by a binary number. And since there are only a few hundred at the most restriction sites on a whole chromosome, we only have to take 8 or 10 individuals to make that binary number a unique signature of that restriction site if the polymorphism frequency is high enough, and I think it probably is high enough. Right? So what that means is if we look into this region from, let's say, a probe arbitrarily on the right, we'll see bands of any particular size as we do a partial digest, but they'll be codable with unique binary numbers, 60, 143, what have you. If we have another probe and we're looking through the same region, again, we look to the right, to the left, we see all kinds of different bands. But if we get this far, then we'll see a pattern in a band size of a particular that will correspond to that binary number, 143. And all we have to do is take enough individuals and have enough polymorphism so that these will really be unique. In other words, what I'm saying is in the limit of a lot of polymorphism, the polymorphism lets you put the map together and actually makes it much more powerful. Now, it would be trivial if this were all there were to it. The one thing that makes this into a non-trivial problem is that in addition to site polymorphism, you can have insertions and deletions. So it's possible that these maps will have register shifts, and then the searches will become much more complicated. Now, that's where we could use your help in dealing with that angle of the problem. All right, here's another case. This shows the best data currently available in separating restriction fragments from mammalian genomes. What you're actually looking at in each lane is a digest of a different species. One of these is human, one mouse, rat, hamster, and so on, cut with a particular enzyme, and separated under the highest resolution conditions that we possibly can. This shows, first of all, very clearly, again, the biphasic distribution of sizes that all of these fragments generate. All of these enzymes generate. But in addition, it shows a very, very discrete pattern of banding sizes. We would love to have the ability to quantitatively analyze this kind of information, because in principle it's providing a detailed fingerprint of a large fraction of an entire mammalian genome. And if we could do a sufficiently good image analysis on these kinds of images, we might be able to use this information directly as a sort of first stage screening for large genomic rearrangements. Now, our effort is concentrating on improving this experimental resolution further, because if we can gain a factor of 2 or 3 more in resolution, we won't need such sophisticated analysis. On the other hand, it's rather difficult, from what I've been told, to actually deal with such images. And I mean, I think we clearly need to interest people who know about Fourier filtering and other image enhancement techniques to try to clean up these images so that one can actually see the patterns. Now, one of the I would say almost unexpected but wonderfully neat uses that DNA technology has allowed in the last couple of years is in paternity testing and in forensics, because it's been discovered that certain sequences in the human genome are so polymorphic that used individually or in concert, they allow you to start to make distinctions among individuals. And it turns out that because we have more polymorphism on our size scale to begin with, that when we start to find such sequences, the degree of discrimination that we have in distinguishing among individuals is truly amazing. And what this slide shows is a probe from a region of the genome called the X-alphoid satellite, which is largely on the X chromosome but has chunks elsewhere. And altogether probably detects something like 30 or 40 million base pairs of DNA from the human genome. So about 1% of the human genome is visible on this slide. And what you're looking at is a family in which this is actually a family of where the paternity is absolutely unquestioned. That's the father, that's the mother, all the rest of the kids. And it only takes one glance at this to realize we can tell everybody apart without any difficulty. And furthermore, there is a tremendous amount of rearrangement going on in the genome, even in this one sort of family. It may be an extreme because this is a repeated DNA sequence, and it may be correspond to a much higher recombination rate than you might expect. With 30 million base pairs of DNA viewed, you would expect to see recombination in something like 30% of the cases, and we're seeing probably maybe 1,000% recombination. So this maybe is a 30-fold hotspot or something like that. But in any event, I think again, one of the challenges that I will toss out to you, this data has to be analyzed by hand at the present time. It's input by hand, and it has to be analyzed by hand. And this particular pattern is very, very informative, and it's really impossible to analyze it by hand. Okay, let me, in the interest of time, move forward and talk just a bit about the sequence management and then sequence analysis. Most sequence data is still entered into the databases today by hand. I hope that will change rapidly, since all the new automated machines have the capability of entering it automatically. The data is read by hand. It's annotated by hand. That means somebody looks at it and says, I think this is the beginning of the gene. I think this is the end. These are introns. These are exons. This comes from such and so. Synonym tables are inadequate. That is, it's not possible in any sort of systematic way to go to the existing sequence databases and ask an intelligent question, like, give me all E. coli sequences. You would think that would be a very straightforward question to ask of GeneBank or EMBL. But in fact, if you do it, you do not get the right answer, because the existing databases don't have an intelligent enough set of synonym tables to recognize necessarily that Escherichia coli, E. coli, various E. coli plasmids and phages and so on, which may be resident in the E. coli genome, are all E. coli. So that's a problem. The ability to link up sequence in the databases, I think, exists, although I don't think it's sufficiently robust yet. And again, there are separate non-integrated sequence databases. I assume in the future, and I think in this case the future is probably within a few months, that the data will more or less rapidly be read, entered into the databases automatically. I hope the annotation will start to be at least semi-automatic. I hope that the matchup and linkage within the database will tolerate imperfections so that we aren't carrying an unnecessary burden of a largely duplicate sequence. The databases should be able to handle horizontal studies. That is, horizontal studies are studies where you compare species or where you compare individuals within a species. And it's very easy to say human genome, 3 billion base pairs, that's a tiny database. But that's not the database, because we have, 6 billion individuals, each of which will differ from the other by probably 1 million sequence differences, so that the database is really quite large in the long run. We must integrate all these databases, and I assume that sequence comparisons will basically automatically become part of any attempt to introduce new data into the database. That is, somebody, at least for the very least, one has to know, is that sequence already in the database, so I don't want it again, or is it almost in the database, in which case maybe I have just the same sequence from a different individual. But all of this really will have to be part of the initial input. Sequence analysis is a very long, complicated slide. I think I got a bit carried away. But my interpretation of what's now possible, this will amuse you, especially if it's very wrong, but maybe it's right, is that we can find open reading frames very well, that we can do this by incorporating codon usage. We can find obvious splice junctions, but we certainly can't find all splice junctions. We know more or less where things stop and start, but not always. We can estimate something about the DNA tertiary structure from the sequence, but those estimates are by and large crude. We can do something about RNA and protein secondary structure prediction, and that most comparisons of sequences are done in a binary mode, that is, sequences taken 2 at a time, which is not certainly the most informative way to do things. I assume in the future we're going to learn how to spot splice junctions much more effectively. I guess for the non-experts, I should just simply say that in many human genes consist 90% of introns and only 10% of exons. And the part you're interested in for most studies is to sort out the exons, those that are really expressed. And errors in detecting the splice junctions can lead to very serious errors in figuring out what a gene is. The control sequences used in mammalian genes are very complicated. And the method of control seems to be combinatorial. And I'll come back to that and explain what I mean by that, because we really need to develop techniques for identifying these control sequences. I presume that reading frame sequences will take advantage of all homology and not just be blind and individual. DNA bending will be much more systematic, and we'll be able to do multiple sequence comparisons on a routine basis rather than just pairwise. Protein structural predictions, I hope, will become practical, but that's not going to be immediate. RNA structure predictions are better and presumably will become more routine. And here's a curious one. We need to know how to identify, when we have it, a sequence that corresponds to a centromere, a telomere, presumably a nuclear matrix attachment site. All these functional elements need to be identified. Well, let me show you some examples that deal with a few of the problems that exist as I see them. This is state-of-the-art sequencing data, as actually obtained by autoradiography. It's much prettier than the example Eric showed you, a small example of one-upsmanship. It comes from really an attempt to systematize and get the highest possible resolution separation of DNA fragments. Nobody has successfully, as far as I know, devised automated ways to read this data by computer with a sufficiently low degree of error. It is the major limitation at the present time, probably, in holding up sequencing of genomes. Because if data like this were computer readable directly, then certain so-called multiplexing techniques, which are very, very efficient, would be usable in generating sequence data very cheaply. But those data require film, at the moment at least, require film output, and so we have to read film. And the problems are obvious that in any film data like this, there are distortions, the lanes are not perfectly straight, the bands are not perfectly square, the data needs to be cleaned up, and the methods for doing this are not good enough yet. Here's a second example. This is more typical sequencing data. This is the type of experiment probably is being done by more molecular biologists. Can we shrink that a bit so people can see the top? You can't zoom this down anymore? Oh, you can see it. It's just I can see it. Okay, this comes from Jerry Crabtree, who's at Stanford. And it's an example of identifying a DNA sequence that corresponds to the recognition site for a certain DNA binding protein, in this case, a protein that is involved in recognizing and activating the transcription of the gamma fibrinogen gene. And you can see, if you look carefully, that when that protein is present, it eliminates a certain amount of the sequence data that's otherwise produced by sequencing chemistry. Right? This is called footprinting. And I think I was recently told that something like 90% of the grants that came into a particular NIH study section in the last round dealt with this kind of experiment. And every one of these is done by hand. Nobody has taken the trouble yet to automate the reading of these images. And there are some very interesting features to it. You can see, for example, that some bands are blotted out, but other bands actually become darker when this particular regulatory protein is bound to the sequence. That probably has clues about DNA bending, structural changes, what have you. And some bands don't disappear. But as we as someone develops the need to process this information automatically, the amount of information that will be generated by molecular biologists and needs to be handled is just truly staggering. Now, this is an example of sequence data. And I love to show this slide. I don't think you need to be convinced, but I show this slide to molecular biologists all the time to point out to them that nobody looks at sequence data. It's a standard pattern in every molecular biologist talk to project a slide like this and say, see, I've done the sequence. And of course, nobody can see a thing because the print's too small and go on. Okay? So I take it for granted that sequences are only going to be looked at by computers, at least at the first go-around. And so this business of sequence management, I don't think I need to convince you, but it's certainly all quite necessary. This is my telephone book example. And the major reason I show it to you is to try to show you how far we have to go. The longest continuous sequence now known, Epstein-Barr virus, about 170 kilobases of sequence, is 12 pages of the Manhattan telephone book. That's all. The E. coli, which will probably be sequenced within a couple of years by the multiplexing methods, is 300 pages. So it's a third of the Manhattan telephone book. The human genome is 200 copies of the Manhattan telephone book. And there's simply no way that anybody's going to start to manage, look at, think about any of this data without a tremendous amount of computer assist. So whether the molecular biologist likes it or not, I think we're in this. It's very necessary for us. And this makes me reflect on something that I think is really amusing. I was interested in computerized methods for comparing sequences more than 20 years ago, when we didn't even have any DNA sequences. And so the best you could do was sort of translate protein sequences back to DNA sequences using the genetic code. And I remember at the time being very discouraged because there was so little data available that you really couldn't do anything. And the problem is really going to be just the opposite, clearly. There's so much data that's going to be available that it's going to be a struggle. Now, I mentioned DNA bending because I am convinced that it's going to turn out to be quite important in understanding probably structure and organization of the genome and probably expression of genes. There's more and more evidence that interesting regions of genomes, like control sequences, replication origins, and so on, are bent. And the ability to calculate this bending is evolving rapidly. It's still in its infancy. This slide I got from Wilma Olson at Rutgers University, who's probably one of the two people that has the most sophisticated algorithms at the moment for predicting DNA bending. And Wilma has been able to show recently that you can make some remarkable DNA structures with particular sequences. This is the most thermodynamically stable predicted structure for a particular DNA sequence, and you can see that it's actually a figure 8. Now, this sequence, I think, is about 1,000 base pairs long. And so those of you familiar with DNA structures realize that this is what we ordinarily call a supercoiled molecule, except the supercoiled molecule is a high-energy form. But in this particular case, the ground state energy, the ground state for the molecule, is already a supercoil. And the so-called relaxed structure, the open circle, would actually be higher energy than this. So it's quite remarkable. I think we have a lot to learn about this. And I would hope this kind of information would start to be integrated rapidly in sequence analysis programs. Here is another case, I think, of a serious need. I just want to call it to your attention. In DNA splicing, in RNA splicing, after a pre-message is transcribed, pieces are cut out, it's very difficult to recognize with absolute certainty where these splice junctions occur, because the local sequence information that dictates a splice site is very tiny, and it occurs far too frequently at random in the genome. And we know now that there's something about the secondary structure of the RNA that is going to be spliced away is sometimes at least very, very important in determining whether an actual splice will occur. Because sometimes mutations out here in the middle of the intron can affect whether or not the splice actually occurs. It's almost certainly occurring at the level of higher order structure. And so we need to develop the algorithms, someone needs to develop the algorithms that will allow one to look at these RNA secondary structures much more effectively to be able to predict splice junctions. Finally, this is an example of the picture which is now emerging for what regulation looks like for genes of higher eukaryotes and even lower eukaryotes. It's very different from the picture that most of us carry around, which is highly based or biased by bacterial studies. In bacteria, most of the available genes in a cell are on, and you need to specifically regulate just to turn off those you're not interested in. In eukaryotic cells, certainly in mammalian cells with 100,000 genes, most of them are off. And what you do is you activate those specific ones that you're interested in a particular cell type. And you do this not by generating a specific activator molecule for each gene. That would be absolutely impossible, because if you have 100,000 genes, you need 100,000 activators, and you get into the problem of who would activate the activators and so on, all right? It doesn't work. So instead, the mechanism that appears to be used is a combinatorial one, in which a set of overlapping tissue-specific, temporally controlled, what have you, factors must bind simultaneously to a particular gene. They all have to be present at once in order for that gene to be transcribed. And so in this way, using a much smaller number of control molecules, you're able to start a gene. Now, what that means as we try to explore the genome is we're going to have to learn the rules of this combinatorial control. We're going to have to learn to recognize the sequences that are bound by these factors, and we have quite a bit of information already. And it's very likely, I think, that the order of these activator sequences can vary tremendously. And furthermore, in many cases, they can be left-right or right-left with no, they can be active in either polarity. And so we're going to need to develop sequence examination methods that allow for various types of permutations and transpositions and still spot the kinds of regulatory patterns which are important. All right. Now, I want to close momentarily. And before I do this, I just want to address three issues for you that I think are important to think about. The first is how complete a job do we need to do in this area? The second is how fast do we need to do it? And the third is what is it going to leave us with when we're done? Well, this is a quote that I love from Sidney Brenner. There's been a lot of discussion in the press and elsewhere that most of the human genome is garbage, so why sequence it? Because most of it doesn't code for anything. And Brenner points out that garbage is something you throw out. Junk is something you keep. And the human genome has kept this. Therefore, 90% of the human genome is junk, not garbage. And people tend to tinker with junk. And probably evolution tinkers with our junk as well. And so most of the sequence in the human genome, which is not coding, is probably not completely functionless. And it may be worth thinking about in a serious way. Now, as for the phasing of the Genome Project, thank you for letting me see this again. Right. Most of us who are heavily involved in this project conceive of it over about a 15-year time scale. Not that that's simply optimum. We think of a 15-year time scale because we can't conceive of doing it any quicker. And most of us can't think any further ahead than that. Right? So it's sort of restricted to that. What I see in terms of technique development over the next 5 years is better ability to clone large DNA fragments, to separate them, and probably the most important unsolved problem is how to take a big piece of DNA and systematically break it up into a series of smaller pieces without losing information about the order of those pieces. And further in the future, in terms of mapping, I see the ability to multiplex map. multiplexing means instead of looking at one thing at a time in a single experiment by tricks, you find a way of looking at many things at a time. In terms of sequencing, new methods are being developed at an amazing rate. There are several commercially available automatic machines which have received a lot of press, but lurking in the wings are lots of other new automated solid-state methods, new types of clever chemistry. There's a tremendous amount of progress which will be made here. In the future, there are people who are actually talking about developing the methodology to sequence single DNA molecules. Certainly that's at the fringe of what one can do in physics. Might be possible. And also, there are other sequencing tricks. Since much of the human genome is repeated, it seems silly to have to keep sequencing the same sequence over and over again, and maybe someone can think of a way to do differential sequencing, that is just start to look for differences between DNA molecules. In terms of productivity, this is what I think most of us feel is likely to occur in the human genome. That genetic maps, within the next 5 years, we'll have, instead of a 10 centimorgan map of the genome, we'll have a map with 5 or 10 times that resolution. I think there's a consensus that that's a high priority thing. It will be done. After that, over the next 10 years or 15 years, I think people will concentrate on using that genetic map together with other data for disease mapping, that is, for finding interesting genes. In terms of physical maps, over the next 5 years, we'll see complete maps of a number of the smaller human chromosomes. Probably within 10 years, we'll have physical maps of all the human chromosomes. And beyond that, we'll start to look at other species. Sequencing, although some people take seriously the notion that we might be able to sequence the entire genome immediately, sort of starting at once on a massive scale, I think a more realistic picture is to imagine that over the next few years, we'll see the production of 1 to 2 million base pair blocks of continuous DNA sequence. Now, I remind you, that's 5 to 10 times the size of the longest block of continuous sequence we have available today. And as time gets longer and our methods get more powerful and more automated, we'll see 5 to 10 million base pair blocks of continuous sequence by the end of the next decade, and maybe the sequence of whole chromosomes in the future. In terms of data and analysis, the part which should be closest to your heart, for mapping, I see the biggest challenge over the next few years is to coordinate all the databases. Let's just have one database that at least scientists can address. And I see the challenge beyond that of making that database friendly to the practicing clinician, because a large number of the applications of that database will be in clinical human genetics. And the people doing those applications don't know any math, don't want to learn any math, don't know much about DNA, but they want to be able to tell their patients what's happening and whether they should seek expert genetic analysis or therapy or not. In terms of sequence, I think the big problem is in the future. It's going to be relatively easy to sort out sequence analysis at the level of DNA structure, probably multiple alignments, DNA bending, RNA secondary structure. That looks easy to me. The problem, which looks very difficult still, is predicting protein structure. Because by and large, we're interested in the part of the sequence that codes for protein. The more likely it is that we can predict protein structure correctly. I don't necessarily mean a priori prediction. Semi-empirical prediction would be fine. But the more likely we can predict what a protein looks like in 3 dimensions, the more powerful will be our ability to compare different sequences and infer function. The ultimate goal, which Eric has already referred to, though, numerous times, and which I'll close with, is that our purpose in all of this is to understand function and not structure. Structure is just a tool that allows us to rationalize and hopefully predict or modify function. And our ability today to predict biological function is miserable. Even if we know the 3-dimensional structure, the complete 3-dimensional structure of a protein, in most cases, it doesn't tell us anything about the function. And I'll close with this example. This is the best one can ever do. It's a very high resolution X-ray structure of a protein. Here, there's no prediction. There's no room for doubt. That's the structure. So I ask you, what does this protein do? Obviously, I picked an example. It's not a very well-known protein. If I showed you lysozyme or ribonuclease, you'd identify it and you'd tell me, well, this is one that's not so well-known. So if you had this, and this is if all our dreams are satisfied and we actually can predict protein structures ab initio from sequence, this is what we're going to get. And would you Would you recommend injecting this into a patient to cure arthritis? Would you eat it for breakfast? What would you do with it, right? It turns out there's scorpion toxin. And I just don't think there's any way that anybody could predict that this blocks a channel, I think it is, from our current state of knowledge. So while we've come a long way, I think we have a long way to go in the future. Thank you.